Hi everyone, in this lecture I am going to talk about capillary electrophoresis. The principle involved in capillary electrophoresis is electrophoretic mobility as well as electroosmotic flow. The electrophoresis is something that is happening under the influence of electrical charge. Phoresis means migration of ions. So electrophoresis actually means the migration of ions under the influence of electric voltage, and the migration actually depends on the sample's ionic charge. If the sample is having more ionic charge, then it will move at a faster rate, and if the viscosity of the sample is less, it will move at a faster rate. But if the viscosity is more, then the migration will be affected, and the particle size is also dependent. If the particle size of the sample is more, then it will move at a slower rate, but if the particle size is less, it will move at a faster rate. And the migration is also dependent upon the electric field. If we apply low voltage, the migration is slower, but if we apply higher voltage, the migration is faster. The principle particularly involved in electrophoresis is electroosmotic flow and electrophoretic flow. So electroosmotic flow is something where the liquid, that is the buffer present inside the capillary tube, will move towards an opposite electrode. It actually happens because of the formation of electrical double layer, that is the surfaced charges that are produced on the surface of capillary tube allows the flow of buffer towards the opposite direction. So the buffer will start moving from one direction to another direction, and within that moving buffer, electrophoretic flow will happen. So electrophoretic flow is something, the migration of ions towards their respective electrodes. If we have a positively charged ion, it will move towards a negatively charged electrode, and if we have a negatively charged ions, it will move towards the positively charged electrode under the influence of electric field. So the separation is happening based on electroosmotic flow as well as electrophoretic flow. And if we have a neutral charge, it will also be finally recovered from the capillary tube, because there is an electroosmotic flow. But in case of other kinds of electrophoresis, the neutral components will not move. But in case of electroosmotic flow, the components that have faster migration rate will reach the detector at a faster rate, but the components with a lower migration rate will reach the detector at a slow manner. But the neutral charges will finally, at last, it will reach the detectors. As you can see, inside the capillary tube there is an electroosmotic flow that is happening. So in case of electroosmotic flow, the buffer, that is the solvent system that is present inside them capillary tubes, are moving from anode to cathode, from a positive electrode to negative electrode. So the solvent that is present inside the capillary tube is actually moving, and electrophoretic flow is also happening. So you can see there are three types of ions present inside the capillary tube. Under the influence of higher voltage, you can see the positive ions are nearer to the cathode. So which means the positive ions are moving at a faster rate, because in the other end we have a cathode. The solvents are moving towards the cathode, and as the component is a positively charged one, while the solvent system is moving towards the cathode, the positive ones are moving at a faster rate, and the neutrally charged compounds are in the middle, and the negatively charged samples are in the are moving at a slower rate, and they are towards the anode side. So for electrophoresis to happen, the components of capillary electrophoresis is the electrodes, positive electrode, that is anode, and the negative electrode is cathode. The ions migrate towards the opposite charges, and the neutral species are unaffected, they remain stationary. Then migration factors. If the samples have a same size, higher charges move faster. Suppose let's say the mixture of components consists of components that are of similar sizes, then how will they be separated? They are separated based on their charge. The components with the higher charge move faster now, and if they have the same charge, let's consider the components have a similar charge. If we have a similar charged components, then the small size components move faster. Okay, the small size components will move faster. You can see that in the diagram given below. On the right side towards the cathode outlet, we have a positively charged components. These positively charged components, first one is a smaller one with 2 positive charge, and the second one is a larger one with 2 positive charge, and the final one is a larger one with 1 positive charge. So based on charges, we have all components are having the same charge, so it can be separated based on their size. So when you see the size, we have a small size component as well as the larger size component, and the small size component will reach the cathode outlet first because of its charge and also because of its size. The instrumentation of capillary electrophoresis include a buffer solution like sodium dihydrogen phosphate, and a sample vial, a source and destination vials, electrodes, high voltage power supply 5 to 30 kilovolt, a sample introduction system, a sample injection, a capillary tube with internal diameter of 10 to 100 micrometer and 20 to 100 centimeter in length, and a detector and a output device. So you can see the instrument consists of a sample vial, that is inlet vial. Inlet vial is otherwise called as a source vial, and on the other side we have a outlet vial. Outlet vial is called as a destination vial, and in between that we have a sample vial. So the inlet vial and outlet vial are kept inside the buffer solution. So if we apply an electrical supply, the buffer solution will start flowing inside the capillary tubes. So the buffer is actually flowing inside the capillary tube, and in the middle of that we have to remove the capillary end from inlet vial, and we have to keep it in the sample vial to introduce sample into the system. And if we introduce the sample into the system based on their separation factor, based on their migration and some other factors such as size, charge and viscosity, the sample components will be separated, and the separated components will reach the detector. Okay, in capillary electrophoresis, the electroosmotic flow is also happening, so it will move towards positive electrode to the negative electrode, and on the side of negative electrode there is a detector. If the components have higher migration rate, they will reach the detector at a faster manner, and they will be detected in the computer system, and it will be recorded as an electropherogram. The applications of capillary electrophoresis is in pharmaceutical industry. It is used for drug purity and chiral separation, and in forensics it is used for DNA profiling and proteins, and in clinical side it is used for microbial detection and for detecting biomarkers, and in environmental side it is used for detecting pollutants, metals and ions, and in food industry it is used for detecting additives and quality testing, and biotechnological industry it is used for proteins, peptides and nucleic acids analysis. Thank you.